for the online students, please, if you have any question, you can write that question in the WhatsApp group, and I will going to request my students that they will going to read your question with your name, so that I can answer those questions. Okay, if you have any question with my earlier classes, last class, we have studied about the members of the specific defense cells. The defense cells who were specific in their actions, remember? T cells and beta cells, and how many different types of T cells are there, how many different types of beta cells are there, how T cells got activated, how beta cells got activated. And we somehow came to know about the MHC TCR complex. Little bit, we started understanding about the intermediate steps. If you know the cascade and their placements right, if you know their placements, it will be much easier for you to decipher the immune system more easily. Placement se murad ye hai ki which of the member is coming first and which of the member is coming after that. That's very important. Like we were studying in our last class, that the beta cell will activated. They have a specific antibody, and if that antibody matches with the trouble, that beta cell is the one which can kill the antigen. That beta cell will further activated, supported by another T cell, which is called as the helper T cell. So that means these two mechanisms were proceeded or were going on at the same time, depending on the severity of the trouble. Okay, please do not left behind in the classroom. It is your absolute right to stop me that I'm not getting whatever you are speaking. Okay, just test me, test my patience. Okay, by asking questions which you want to try. Don't worry, इससे कुछ नहीं होगा. You just made a statement and I did not get anything out of it. Please repeat that thing or try to use an alternative version to explain to me or explain to us. That is absolutely fine to ask questions, to stop me. It is absolutely fine to go slow in molecular immunology, because we need to stick around one particular thing. And now you can start feeling that the heat, which I explained you in your first class, that we will going to talk on the same things again and again. But like every time we will go further deep into the concepts. And that is the path I have selected for you. I believe this might be the best way. General understanding, more specific understanding, more close understanding, and the understanding at the molecular level at the end of the day. So we are just, I mean, imagine I introduce you about there is an interaction between the MSC and the TCR complex, which communicate that information from the macrophage to the T cell. And imagine a chapter is all about MSC and TCR interaction. Only about MSC. So we will be looking at the protein structures. We will be looking at how MSCs were created, how they able to capture the antigen, and how they able to present that antigen on the surface. What are the factors which actually defines that this process will be executed in a perfect way or not? Further detail. So it's like knowing about the things, looking closely about a thing, and then analyzing the same thing at a molecular level. That's what we will be doing in the molecular immunology class. I wish that this journey will going to help you to create bigger things. I want you, as I told you, that I want you people to create proposals out of those understanding, to create proposals out of whatever you are listening to me. And in order to have a better understanding or understanding at a undergraduate level as compared to international arena, I'm not talking about in comparison with your friends, you should have an idea about these things. And let me start. Okay, here we go. So last time we were like, this is the slide. These are the cells which give rise, which produce most of the cells, all the cells related to our immune system. And all those cells which are the major player of our immune system, all those cells, they came into existence from a common source, from one cell. And that one cell is called as the hematopoietic stem cell. Before I have explained what are the stem cells, what is meant by the hematopoietic stem cells, they are not able to produce everything. बोला था ना मैंने, they are not able to produce everything, but they are able to produce a certain category of cells. They're called multipotent stem cells. Hematopoietic stem cells are multipotent stem cells. They are on a similar sort of stem cells are present in different parts of our body, and those stem cells are called as adult stem cells. Because in different parts of our body, we need a constant supply of new cells. Imagine skin cells. They are constantly, we are losing the skin cells at a constant base, on constant basis, regularly, because of the physical, wear and tear. We are losing the skin cells from our body, and we need to replace those skin cells constantly during our lifetime. So nature has placed certain stem cells over there, which are capable not only to maintain their own numbers, but they also give rise to the skin cells constantly. Similarly, nature has given us the hematopoietic stem cells, which are present in our bone marrow. But I want you to think earlier than that, before that bone marrow, when we first born, during the development, before we born, during the development. At that time, it was created at different places. At that time, the spleen and the other organs are involved in that, in hematopoiesis. But once we are born, the hematopoietic stem cells, they are present in the bone marrow. And they are the ones who are producing all sorts of cells which are constituting the immune system. What are those cells? Look at these. At the bottom, the beta cells, the T cells, neutrophil, monocyte, eosinophils, basophil, platelet, erythrocytes. All these are the cells which are constituting our immune system. They are fighting against the bad things one way or another. Their jobs are different. Their mode of actions are different. They are working under different conditions in different scenarios, but all of them belongs to one army, and that is our immune system. And all of them came into existence from a common source which is called as hematopoietic stem cell. And the process is called as hematopoiesis. So hematopoiesis is a constant differentiation phenomena. Differentiation, and the cells are dividing as well as changing their morphology. Cells are dividing as well as changing their morphology to produce all those combination of cells. A differentiated cell do not have the potential to become everything, do not have the potential to become something. They are one definite cell with definite function. So hematopoietic stem cells, they have the maximum potential of becoming many things. They have the maximum potential to become all sorts of immune cells. But with the passage of time, when they trigger the process of differentiation, their ability to become many cells decreases. And you can call it as stemness. The level of stemness they are carrying, it starts decreasing. And in return, they will receive more differentiation. More differentiation. The cells become more differentiated. So the loss of stemness give rise to the differentiation. A differentiated cell is the one which is capable of executing a particular job, which has definite proteins, structures which can execute. T cell is a differentiated cell. Beta cell is a differentiated cell. Eosinophil is a differentiated cell. Blah, blah, blah things. Hematopoietic cell is a stem. It is not able to produce any function. It is not designed to do any job. It is designed to give rise to also all those cells. Stemness, loss of stemness, give rise to the differentiation. A similar sort of thing occurs during the process of development. Development में भी ऐसे होता है. The zygote has the maximum capacity to become everything. 2 cell stage, 4 cell stage, 16 cell, 200 cells, 1000 cells. Then they started losing their stemness because they started getting differentiated. This is very critical. Now, the level of stemness decreases or changes. If you ask me, so all these cells belongs to one thing. All of these cells, they like have a common origin. All of these cells have a common origin. So that means they should have a similar sort of, I must say, genotype. They should have a similar sort of phenotype. They should be closely related to each other. But they are different. They are executing different things because the patterns of expression of the genes they are carrying, it is different. So hematopoietic stem cells means that the cells are using the combination of genes in a different way. When they started becoming differentiated, they will start using a different combination of the genes. That does not mean that these cells are carrying different types of genes. They are all carrying the same DNA, same sequence of, and the same number of genes. But they start selecting the different pattern of expression of the genes. That is why they started becoming different. If you ask me why the first member is different from the second member, the second layer is different from the third layer, on what basis? Based on the difference of the expression patterns of certain genes that will started becoming, they started pushing them to become different. मैं इसको explain करता हूँ. For example, it is like the piano keys, which are like having same piano keys, same gene sequence, same genetical material, but you can strike them in different combination to produce different tunes. So the cells utilizes the same genes in different combination to produce different tunes, and they started becoming different. As long as their stemness is maintained, they can become all sorts of cells or many types of cells. But once they started becoming differentiated, they develop and adopt definite structures, and then they are ready to perform and execute a specific job. G. I love the analogy because Mustafa akha kithe jaal rahi hai, kithe read te kithe tera ye. But that is every living form at a cellular level, if different from each other, it is because they are using a different pattern of expression. Full stop. It is that much general. Now the problem is that who is responsible for this different type of regulation? अगर आप मुझसे पूछें, I don't want to make you more, don't want you to take you the more interesting side of the science. Just stick with the boring stuff. If I ask you, the cell of the rods and the cones in our eye, they are differentiated cells. They are designed to produce definite functions to capture the sun, to the light, and then interpret the light and send the signals to the neurons and blah blah. Think about the cells of the skin. Differentiated cells designed to execute definite functions. You know? If I ask you that whether the skin cell is carrying the genes which are related to the vision, what will be your answer? Yes or no? Yes. You know? If I ask you whether the eye cell, rods and cones, the eye cells, whether they are carrying the genes which are related to the skin, the answer is yes. So that means every cell contains all the source code, all the library, all the material required to execute a complete life. But the rods and cones are acting like a rods and cones since they are not using the genes which are not related to the vision. And that is why they will start becoming different. The skin cells, they just using the genes which are related to the skin, but not the. It is that much simple. जब पहली बार इसको किसने सोचा था ना, क्या ज़हन में आया होगा इस बात से कि a skin cell is carrying a full life. It does carry the genes which can become an eye cell. It does carry the genes which can make them like a kidney cell. It does carry the gene which can make them like a stem cell. It can become like everything, but somehow it is not using all those genes. If you understand the mechanism of differentiation, one of the best area of research, I can tell you, gene regulations. Life is not about having DNA. Aha. Life is about how you can control the DNA, how you can regulate the DNA. Otherwise, आप एक sequence of DNA लगा के किसी बोतल में बंद कर दें, DNA बना के खुद बना सकते हैं हम. You know, you can create DNA nowadays by yourself with machine. ह्यूमन जीनोम बना के डाल दें किसी मशीन के अंदर, वो इंसान नहीं बनेगा. किसी बैक्टीरिया में डाल दें, वो बैक्टीरिया इंसान नहीं बनेगा. It's about how you can regulate the genes. So somebody thought at that time, if the skin cell is carrying all the source code of life and it is just not using it, just using only the part which belongs to the skin, how about requesting the skin cell to please use the other portions, the other genes, to start using all the genes, start making available all the genes, then what will happen? The skin cell will become a different cell. It's no longer a skin cell. If we able to control this thing more in a better way, and you push the skin cell and request the skin cell to start using the patterns of expression in a way a stem cell is using, the skin cell will become a stem cell. And we made it. It is called induced pluripotency or induced pluripotent stem cells. A stem cells who were not supposed to be the stem cells, but we push them to become like a stem cell. यामानाका ने ये काम किया, Nobel Prize win, कितनी बड़ी बात. And the name of the article at that time, it was published like with a very wonderful heading at that time, Going Back in Time. बड़े साइंसदान भी कभी कभी बड़े आर्टिस्ट होते हैं, कुछ साइंसदान लिखारी होते हैं. I mean, I was very much impressed with that title, Going Back in Time, converting the skin cell into a stem cell, going back in time. पता कैसे? लोगों को ये भी गलतफहमी होती है कि साइंसदानों के क्रश नहीं होते. उनके होते हैं. They are normal people यार. आप मैरी क्यूरी को देखें कभी, रोज़लिंड फ्रैंकलिन को देखें कभी, अपनी आंखों से नहीं, किसी साइंसदान की आंख से देखें. Beautiful people. So the Yamanaka used that phrase because if you imagine the process of development, it's like fully capable stem cell becoming everything. यही होता है ना, baby become everything. All sorts of cells coming from one cell. And then it started dividing and differentiating. In the meanwhile, it loses its stemness, it receives the differentiation, started developing new cells, started developing new organs, tissues and new organs. So the skin cell is a future version of a stem cell. है ना? पहले stem cell आया और उसके बाद वो करते करते क्या बना? If you push the skin cell to become a stem cell, it will be like going back into time. Time machine है, biological time machine. Amazing. जी.